Hi, my name is Omar Gina, Chief Sales Officer of the Blacktrace Group of companies. And Blacktrace is the parent of a number of different companies, including Syrris, leaders in batch and flow chemistry, Dolomite, innovators in microfluidic and droplet technology, Glass Solutions, experts in scientific and architectural glassware, and also Centeo, who have some temperature control modules for biological applications. So I'd like to introduce you today to Max Drobot, Head of Applications for the group. And Max is going to teach us a bit about flow chemistry. So, Max.
Thank you, Omar. First, a very quick word about Syrris. So Syrris is a company based in England, in Cambridge, which design, develop, and manufacture equipment for R&D chemists with a strong focus on automation and ease of use. Our product cover batch chemistry and flow chemistry. So, all of us are familiar with batch chemistry. That's how we were taught chemistry, and that's how we're still doing chemistry for most of us. The type of glassware we're using are like round bottom flasks, such as this one for very small scale, or for larger scale, we can go onto jacketed reactor, such as this one. In flow chemistry, we've got a different approach to how you would do your reactions. And instead of using standard reactors, people are using what's called a microreactor. Those microreactors can be glass microreactor, such as this one, with etched channels of about 100 nanometer diameter, or you can also use tube reactors with the length of fluoropolymer or metal tube wrapped around a heat exchanger. Or you can also use glass columns, such as this one, which can be packed with a solid and enables you to handle solids as well as liquids in flow chemistry. So what's happening in those reactors? What is flow chemistry? Well, in flow chemistry, the key thing is everything is continuous. So reagents, starting materials, and reactants are continuously pumped into a microreactor where they mix and are exposed to reaction parameters such as temperature. The reaction happens, your product forms, and the product leaves as a continuous stream. So compared to batch, which is a traditional way of doing chemistry, where you load everything in the reactor, then start heating, start mixing, your product form, and then to recover your product, you have to stop everything and do a workup and filter. While in flow, your reagents are continuously pumped and the product leaves as a continuous stream. So it's a continuous process. It's not a start and stop process such as batch. Reaction parameters for batch are typically temperature, mixing, reaction time, and concentration. In flow chemistry, key reaction parameters are very similar, but with a slight difference. So instead of reaction time, people are talking about residence time. Instead of concentration, people are more interested in molarity ratios. And temperature is still very important. Mixing, as we're going to see in the next few slides, is not as critical, and instead people prefer to focus on pressure. So let's talk about the key reaction parameters in a bit more detail, starting with the residence time. So what is residence time? Residence time is how long every fraction of your reaction mixture spent in the microreactor, from the point where it's mixed and exposed to the reaction conditions up to the point where it leaves the microreactor. The formula to calculate it is given by the volume of the reactor divided by the flow rate. And already that formula enables us to see that there are two ways of controlling the residence time in your flow chemistry experiment. The first one, the obvious one, is to change the size of your reactor. So you can do a reaction on a small scale like this one, and then if you want to increase the residence time, then you can increase the volume of your reactor and move on to a tube reactor such as this one with a volume 4 times, 16 times bigger, and your residence time will increase accordingly. The second way of controlling the residence time is by changing the flow rate. And for that, you need a very accurate and smooth pump, such as a Syrris Asia pump. If you increase the flow rate, then your residence time will diminish. If you slow down the flow rate, then your residence time will increase. And also, not only can you play with residence time, but you can also play on the ratio between your different reagents. So, for example, at the same concentration, if you pump one reagent twice faster than the other one, then you're introducing twice the amount of material in your microreactor, and your ratio will be 2 to 1 in terms of molarity. Talking about the other key reaction parameters, I mentioned mixing. So mixing is not as essential as it is in batch. The reason for that is because we are in laminar flow conditions. Because of the very small channel diameter, flow conditions are laminar, which means that the mixing is diffusion limited. And because of that, it is highly reproducible and ultra fast. Another key parameter is temperature. So again, because of the very small volume of internal solutions, you've got a high surface to area ratio, and the heat can be brought very quickly into your reaction and can be dispersed very quickly as well. So you've got a high heat transfer, and because of that, the temperature control is usually much better than what you have in batch. The final one is pressure. So I mentioned that people like to pressurize the flow reactors, and the first reason why they want to do that is because it's very easy to do. The reactor volume is so small that there's no really risk in pressurizing it and compared to batch. And when you do that, when you pressurize the flow reactor, you can heat your solvent above its standard boiling point. It's called superheating. And you can also handle gas and liquid together and make sure that the gas diffuses in the liquid phase. The basic simple flow chemistry is between two miscible liquids. You can also have heterogeneous flow chemistry using solid and liquid. By example, by using the column that I showed earlier on, if you pack that with a solid reagent and flow a liquid reagent through that, then you have a heterogeneous experiment. You can also have gas liquid and combine the three to have gas, liquid, and solid. And you can also have immiscible fluid like aqueous and organic as well, which will form little reaction slugs, and the reaction will happen at the same rate as we will have in batch. So why do people want to do flow chemistry? Well, they want to do flow chemistry because of a range of benefits that they can only see in flow. Things like faster reaction, safer reaction, more control overall, so like reactions that are cleaner. You can also do very fast reaction optimization. Once one of your experiments has left the microreactor, then it only sees solvent, it's clean, and the next experiment can start. So it can scan a wide range of reaction conditions very quickly. And things like integration of analytical device or workup are very easy to achieve as well. So I want to finish with an example of chemistry that has been published by a renowned group, which is Steve Lay's group from University of Cambridge, who are world experts in flow chemistry. They published a paper that demonstrates the synthesis of a pharmaceutical active ingredient starting from very simple starting materials. The full process is done in 7 steps, and it's all in one continuous process. There's no workup in between, no stopping in between. And they exploit all the benefits of flow chemistry. Superheat dichloromethane to 80 degrees, use gas, liquid, and solids. All these in one continuous process. So I hope that this short talk convinced you that flow chemistry can be very beneficial for your process and for your science. And Syrris is a world leader in flow chemistry with systems like the Syrris Asia flow chemistry system. And if you're interested in finding more about it, please go to www.syrris.com.